Our guest today is Professor Julie Andersen, postdoctoral fellow at Harvard Medical School and senior scholar at Buck Institute on Aging. Welcome to our broadcasting room, Professor Andersen. Our audiences can't wait anymore to listen to your share. Then first, could you please make a brief self-introduction to the audience?
Sure. So I received my undergraduate degree in biochemistry from Washington State University. I did my PhD at UCLA, which is when I initiated my studies on Parkinson's disease, spent a postdoctoral fellowship at Harvard, Department of Neurology, was at my first faculty position was in the School of Gerontology at USC. And 22 years ago, I joined what was at that time the newly opened Buck Institute for Research on Aging in Novato, California. And as we'll discuss, the emphasis of my lab is really on understanding what role aging plays in Alzheimer's and Parkinson's disease.
Thank you, Professor Andersen. Now, before starting the interview, we noticed your involvement in the recent breakthrough research, taurine deficiency as a driver of aging, which was published on Science. Then we'd love to know your expectations for the clinical application of taurine, and will you also participate in its clinical research?
Yeah, so as this audience likely knows, so taurine is a naturally occurring amino acid. There's an age-related reduction in its level, and that's been linked to a number of different chronic human diseases, including type 2 diabetes, hypertension, obesity. And in this, I think, really groundbreaking study that, as you alluded to, was recently published in Science, the senior investigator, Vijay Yadav, who I see you interviewed fairly recently, had the idea to look at the impact of taurine supplementation and whether supplementing taurine would be able to restore youthful levels of the compound and what effect that had on the lifespan. And what I think is really nice about this study is that it was looking across very disparate evolutionarily different species, all the way from C. elegans, which was the work that the Buck contributed to. Was a very talented research scientist, Manish Shamoli, who was intimately involved, all the way up to non-human primates. And also, interestingly, the supplementation was found to affect a lot of different hallmarks of aging, including things like cellular senescence, telomerase deficiency, mitochondrial dysfunction, DNA damage, inflammation, and it corresponded with improved function of various organs, including bone, muscle, brain, fat, gut, immune system. And so it really raises hope that simple and safe interventions like this could have a huge impact on aging. And I also think that it serves as an example of the strength of interdisciplinary research. This study involves something like, it's over 50 authors across 34 different institutes.
It's a huge research.
Yeah, huge study, which I'm very excited to have been a small part of. While I think that the results do call for double-blind controlled clinical trials, I think we as authors have been very careful to caution against people self-dosing until those trials are done, and also to realize that there's a lot of different ways to boost levels of the metabolites, which include diet. Taurine's found in foods like shellfish and dark chicken and turkey meat, and also bouts of exercise. So there are, in essence, kind of natural ways to boost taurine levels as they dissipate during aging. So very cool study. And I give all kudos to VJ for this wonderful work.
And 2 weeks ago, we welcomed Professor Yadav to our broadcasting room and have a chat with him. He also expressed the same point of view and expectations as much as you. And we're also looking forward to the breakthroughs in the clinical trials of the taurine deficiency as a driver of aging.
Yeah, I don't know how much the buck will be involved in those per se, but I know that there are ongoing studies at the preclinical level that are looking more towards drilling down on the exact mechanisms that are involved. I think that's important because by understanding mechanisms, you may identify further players which are involved in the process and which lead to additional therapeutics that sort of mimic what taurine is doing and maybe more effectively. So lots to be done.
Looking forward to that. Now, shall we dive into our theme of today, that's the aging of the brain? Well, since that our audience are normal people, but not professional researchers, then could you please briefly introduce the neurodegenerative diseases to us first, such as the Alzheimer disease, Parkinson disease, then you may include the symptoms, risks, or vulnerable populations?
Sure, absolutely. Love to. So starting with Parkinson's, traditionally Parkinson's has been characterized by loss of voluntary motor movement. And that loss is due to death of a set of neurons in the midbrain in this region called the substantia nigra. And these neurons contain the neurotransmitter dopamine. And these cells are part of a larger neuronal circuitry which controls motor movement. So you have this sort of electrical circuitry, and when you lose these dopaminergic neurons in the substantia nigra, it's like you've cut through the wire and all the lights go off. It's interesting that this term substantia nigra is due to the fact that these neurons, which contain dopamine can be converted to a type of melanin called neuromelanin. And as a consequence, the neurons appear black in appearance on, postmortem autopsy. And if you compare the brain of a Parkinson's patient to that of an age-matched control, you can actually see the reduction in levels of these neurons. The disease also includes buildup of this protein called alpha-synuclein. And this was really discovered, wow, I can't believe it, but like over 20 years ago, where people discovered that there were some rare familial cases of Parkinson's disease. And when they isolated the mutation in this family, it turned out to be in a gene that encodes this alpha-synuclein protein. And then when people went to look at people who have what's called sporadic Parkinson's, where there's no genetic mutation or familial component, they found that within the neurons of even people who don't have an alpha-synuclein mutation, they have a buildup of this alpha-synuclein protein. And so over the course of the last couple of decades, that's been a major topic of interest for researchers in the Parkinson's field. Because you're affecting this voluntary motor movement, when you lose these neurons, patients experience symptoms like freezing or tremor, loss of balance. It's usually in sporadic cases, it's initiated like around 65 to 70 years of age. It's interesting, it tends to be more common in men than in women.
Yes, many of our audiences also reported that their parents are suffered from these diseases and also have these symptoms or indication.
Yeah, I mean, somewhat ironically, my mother, who's in her 80s, was diagnosed 3 or 4 years ago with Parkinson's disease. And as we may get into, she, like a fair number of Parkinson's patients, experienced gastrointestinal issues 10 to 20 years before she was diagnosed with Parkinson's. And that is a not uncommon feature of PD. So
sorry to hear that.
Yeah. So there in the last, a couple decades, there's been a lot of emphasis on looking at the genes involved in Parkinson's. Not that these gene mutations are responsible for many of the cases of PD, only maybe 10%. But the thought was that by understanding what if mutations in these genes are causing a problem, then it kind of points to particular pathways that maybe even when they go awry, play a role in sporadic cases of the disease. So there has really been, with both Parkinson's and Alzheimer's, a lot of emphasis on genetics. But for those of us studying aging, we realize that genetics really only contributes to maybe 10% of aging. And so there's a growing recognition in aging itself and in age-related disease, a growing appreciation of the role of environment. And Parkinson's disease is something that's been known and studied for quite a while. There's a strong link with environmental factors, including exposure to certain pesticides like Paraquat or rotenone, which is something which are used to rid crops of what do I want to say, of things that would be harmful to them. But there is this very strong epidemiological connection between, for example, a farm worker's exposure to pesticide and later developing Parkinson's disease. In fact, it's probably one of the environmental toxins which has the strongest tie to a disease state. And that's led to use of toxins like Paraquat in preclinical models to study Parkinson's disease. It's also, so there's this risk with the environment. I think in terms of Parkinson's disease, and I think we more and more think of this in terms of many, many different age-related diseases, but there's kind of this triple threat, which includes not just aging. A lot of these chronic diseases, the older you get, the more you're prone to present with these. There's also genetics, and it may not be a familial mutation, but there may be genetic predisposition. For example, with cardiovascular disease, people may have certain alleles or certain forms of genes that affect, lipid metabolism. So they're going to be more prone to having cardiovascular disease. And then if you couple this with no exercise, poor diet, stress, this all kind of works together to contribute. So it's kind of this triple risk factor: aging, genetics, and environment that we really think contribute to a lot of this.
As you have mentioned, that aging itself and aging-related diseases may play an important role in neurodegenerative diseases. Well, can we clearing the senescent cells using, for example, senolytics and other drugs can effectively prevent brain aging and other neurodegenerative diseases?
So senescence has been, I'll tell you a story. I was part of a grant from the NIH about 15 years ago that involved somebody who's very prominent in the senescence research field, Judy Campisi. And this grant was really looking at how the mitochondria, these power plants in the cells that produce needed energy for all of the cellular functions, we're really looking how mitochondrial dysfunction contributed to aging and various age-related diseases. And during the course of working with Judy as part of this grant, I sort of turned to her and said, it seems pretty clear that that cells in the periphery, when they undergo senescence, this can contribute in various tissues, contribute to disease, but it doesn't seem like many people are looking at the brain. The dogma at that time was that senescent cells had to be replication competent. And I think when people think about the brain, they usually think about neurons, which are post-mitotic. But in fact, the brain has a number of cells which support cells, glial cells, which are actually able to replicate and will replicate under stress conditions. So interest over it's really just been in the last 5 years, interest has really become very fierce in looking at the role of senescence in Alzheimer's and Parkinson's disease. And in fact, as you and your audience may know, use of senolytics as a treatment for Alzheimer's is currently under clinical trials, including at Wake Forest. I would, based on our research, make a note of caution in that when you're giving senolytics to a multicellular organ, which most organs frankly are, there may be an issue with finding a dosage of the senolytic that's going to selectively remove the senescent cells without impacting on healthy cells. So in our laboratory, in long-term human brain cell cultures, our data in treating these cultures first producing Alzheimer's in a dish in these cultures by giving them A beta oligomers. These cells, both the glial cells and the neurons, undergo senescence. And if we treat with a senolytic, we find that, for example, while cells like senescent glial cells, astrocytes, thymic glial cells, may respond to a particular dosage of a senolytic in the desired manner, you get selective killing of senescent glial cells and not other cell types, it may actually have detrimental effects on healthy cells like neurons and could kill those cells. So I think that's an important consideration. And one of the things that we're interested in our lab is developing more targeted approaches to administer drugs like senolytics using genetically engineered cells like immune cells. And the idea is that you can develop, so we're trying to develop systems, cellular delivery systems, where the immune cell will only, it will travel to a site of pathology, for example, in an Alzheimer's brain, like an A beta plaque. It is genetically engineered to produce drugs like senolytics that can prevent loss of, let's say, neurons. And then once the A beta pathology has been removed, then the system turns off. So I think we need to be really thinking about how we're going to target drugs, because right now, even for things like immunotherapy in Alzheimer's disease, you're giving an antibody against A beta. You have to give a really high dose antibody for it to cross into the brain. And it's not only impacting on the plaques and tangles, but because it's bulk drug delivery, it's going to impact on healthy cells as well. So that is, in fact, we just recently received an NIH transformative award grant, which is, these are kind of high risk, high reward to test these kind of cell-mediated drug delivery systems. So we're really excited about that.
Your research has mentioned that a Paraquat induces cellular aging and leads to cognitive diseases. Well, are there any other substances may cause the similar effects?
Yeah, so Paraquat, as I mentioned, is this pesticide, which frankly has been banned in a lot of European countries, but is still in use in the US. There are these epidemiological studies that show that there's an increased risk for development of PD in people that have been exposed, as well as preclinical models like worms, mice, non-human primates.
Well, it
And
seems that Paraquat is not forbidden in China too.
yeah, or Brazil, actually. It's very interesting. In fact, there have been some lawsuits brought against some of the manufacturers of major manufacturers of pesticides involving some of these farm workers that that developed. Yeah. So as you alluded to a few years back, and this is one of the first studies to look at a model of Parkinson's disease, we found that so it's known that if you give Paraquat at a certain dosage to mice, they develop Parkinson's disease. You get loss of these midbrain dopaminergic neurons, you get increase of alpha-synuclein, the mice show motor dysfunction. And we found that if we genetically eliminated senescent cells, we could prevent development of Parkinson's disease in that model. And that paper was published in 2008, early 2018 in Cell Reports. And then this field has kind of exploded since then. On the heels of that, there were a couple papers which came out looking at different preclinical mouse models of frontal temporal lobe dementia, which is related to Alzheimer's disease. So there's been a huge interest in senolytics. In terms of like how does Paraquat work, it's known to be a redox cycler. So in other words, it produces oxidative stress, which can kill vulnerable neurons. And the levels of oxidative stress are known to be increased in brain regions in PD patients. And oxidative stress can induce senescence. So that may explain why there's an increase in levels of senescent cells, both in postmortem Parkinson's brain tissues in the substantia nigra and when you give mice Paraquat as a model of PD. There are a number of other substances that are known to emulate features of PD. I mentioned rotenone. Rotenone is something that has been used to call fish populations within lakes. It's actually an inhibitor it's a mitochondrial inhibitor, so it actually acts to prevent this organelle within the cell from producing energies the cell need to function. And if you give rotenone at a particular dosage regime to mice and rats, they show signs of Parkinson's disease. MPTP, which was something that was discovered in the late 70s, really by dint of the fact that there were drug addicts in the Bay Area in San Francisco that were trying to make, in essence, a heroin analog. So they ended up making this compound MPTP that when they injected it, and it's very sad because these were young men, and they ended up with kind of an acute Parkinsonism. And people again realized that this was emulating what you see in PD, and it was used for several years as a model in mice Parkinson's.
That you have mentioned the mice, they will throw up symptoms or implications of cognitive diseases. Did the researchers raise them with any other drugs that may lighten their symptoms or treat diseases?
Yeah, so a lot of before we really understood some of the genetics behind familial Parkinson's disease, the models that were used were really these, what I would term neurotoxin models, where people would for example, give a mouse MPTP, and the mice would sort of have this acute development of phenotypes, neuropathology, behavioral, motor effects that you see in Parkinson's patients. And these were actually used to test different therapeutics. The downside to those neurotoxin models is they tend to be, unlike what happens in Parkinson's disease, which is development of the symptoms and the pathology in an age-related manner, and also kind of slowly. These animals would develop Parkinson's very rapidly. Even in young animals you could produce. So like for myself, it was somewhat problematic in that you're not really then looking at the contribution of aging. So I will say for these original senescence studies, we did use Paraquat. In a way, it was because it is rapid. The pathology is due to production of oxidative stress. Oxidative stress is known to produce senescence. Since that time, a colleague of ours, Juan Kim, at University of Delaware, has looked at senescence in the context of more chronic models, like genetic mouse models that are expressing human forms of mutated alpha-synuclein. And they've also, in those models, seen that this increase, you have an increased presence of senescent cells. I think we're still trying to sort out in the AD and PD research communities what cells undergo senescence in these diseases, and does it different from, in a sense, although we talk about Alzheimer's and Parkinson's disease, these are probably syndromes. Different people present in different ways. And again, it's very complicated because it's a mix of not only increased risk with aging, but also as individuals, we all have a particular genetic makeup. We all have particular lifelong environmental exposures, and that can all, in a kind of complicated way, contribute to whether someone's going to end up presenting with these diseases. So I can say in some of both the AD and PD models, depending on the model, you see different cell types undergoing senescence. So I think a lot of times biology, we start out, especially for people like myself that are sort of molecular biologists, we start out with very simple hypotheses, and biology is complicated, so things start getting a little more complicated as we look at them a little bit more. But a very exciting advent, because so much emphasis had been put on plaques, A beta plaques, and neurofibrillary tangles that contain this protein tau. Those were kind of the two neuropathological hallmarks that are associated with Alzheimer's disease. And a lot of the research over the last 20 years was towards making antibodies that would eliminate those pathologies. And until recently, clinical trials using that type of immunotherapy wasn't very successful. There have now, and over the course of last year, been a couple of trials that seem to suggest that immunotherapies against A beta plaques actually seem to slow cognitive loss. The flip side to that is that because those immunotherapies are bulk and they don't just target the plaques, they can also target things like the vasculature in the brain. And so a serious side effect of use of these type of immunotherapies is it can cause brain bleeding. And that can lead to death in a subset of the patients, which is another reason why I think it's, very important that we think about how to target these medications to the pathology. So yeah.
And the research has suggested that the APOE4 gene increases the risk of dementia, includes Alzheimer's disease, and about 25% of people actually carry this gene. It's really a large percent. And how can people already carrying this gene avoid cognitive decline? Is there any method?
Yeah, so with, as you say, these ApoE4 carriers have a higher risk for developing AD, and it's particularly bad for people who have two alleles, one from the mother, one from their father. You can be tested, so you can take a genetic test to figure out what your ApoE status is, and if you're an ApoE4 carrier. To date, if you look at recommendations for ApoE4 carriers, they really include lifestyle recommendations like diet, Mediterranean diet, fresh fruit, veg, nuts, oils, fish. There are other things that are kind of, I mean, it's really a matter of I have this gene, I'm more susceptible, so I should try. I can't control my genetics, but I can control my environment. So I can control things like my diet. Also, hearing loss has been shown to be something that contributes to dementia. Getting your hearing checked regularly. Not smoking, keeping your mind and body active, things like diabetes are so people who are type 2 diabetic have a higher risk of Alzheimer's. Not is particularly associated with ApoE4. So,
and do you think
if you're
it is meaningful for us to have a detection on this gene? If I know that I have this gene, it is.
I think it is, because I think you can. I would love to say that we right now have a magic bullet that if you have this gene, you could be given a pill that, but at the moment, it's really about controlling environmental factors that can contribute. You wanna not end up with diabetes. So you want to not smoke, you want to eat a good diet. Other things associated, especially with ApoE4, are, obesity, metabolic disease, hypertension, hyperlipidemia, not getting regular sleep, all of these things. It's interesting because it turns out that a lot of these immunotherapies that have been, that look like in these recent clinical trials, have been effective. They seem not to have been effective, although it's a small number that have been tested in ApoE4 patients. So we really need to do larger studies to make any meaningful conclusion. But I do think genetic testing is useful, because the earlier your risk is diagnosed, the earlier you can adhere to the things you can do. And, hopefully the earlier, and I think we're in a kind of a watershed moment, the earlier then you would be eligible for trials using things like immunotherapy or senolytics or whatever. So I think it's important, both in terms of prevention, preventing developing it, just doing the things you can do, and also keeping an eye on the development of novel therapeutics, which I would say over the last 5 years has been pretty rapid, which is promising.
Yeah, as long as we know that we carry this gene, we may
Exactly.
focus on good things that may help us.
Right, exactly. Yeah. I mean, the sad thing about things like Huntington's is you can take the test, find out that you have it, and there's not really a lot of lifestyle changes that are going to slow the course. So at least there is that. I think with both Parkinson's and Alzheimer's, there is that ability to both pay attention, make sure you get regular health checks. And really it's like a lot of the things which can contribute to development are really other age-related diseases like diabetes or hypertension or whatever. And I think sleep, as we've found out in the last decade or so, sleep's.
You have discovered that the benefits of urolithin A, a metabolic genetic from the allergic acid via its metabolism by the gut barrier from the brain. And then moreover, you have mentioned that in the case of Parkinson's diseases, it actually starts in the gut. So therefore, is there any new evidence suggesting that enhance the gut microbiome that can protect against these neurodegenerative disease?
Yeah, there's actually, and it particularly with Parkinson's, there's kind of a growing body of evidence that suggests that things that enhance gut function, and that can, besides neurons in the brain. But as I sort of mentioned, there are patients who have 10, 20 years before they start showing motor dysfunction associated with PD, they have gut issues. So they might have inflammatory bowel syndrome, constipation, etc. And that led people to look at the microbiome in Parkinson's patients versus age-matched controls. And it turns out the microbiome, and above that, it looks like that we may actually produce alpha-synuclein, this toxic protein in the gut, and it can travel across what's called the vagus nerve up to the brain. And so there's been a lot of interest in terms of what can you do if it starts in the gut, what can you do to prevent the spread? And I will say recently, I think there's been growing body of evidence this may hold for Alzheimer's disease. The number one best medicine is especially high intensity, but also things like yoga and dance. Urolithin A is a compound which is produced by bacteria in the gut. So it gets converted to urolithin A, and people taking this orally, we showed in a preclinical model of Alzheimer's disease that actually giving mice this urolithin A orally actually reduced pathology and prevented cognitive loss.
I remember that the mother of Elon Musk, her name is Mayor Musk. She also take the urolithin A supplements.
Yeah, there is you can buy it as a supplement, and there are some small clinical trials of urolithin A in humans where they showed that if you take kind of older, healthy, sedentary adults, and you dose them, or they take urolithin over the period of a few weeks, they actually show improved motor function. And I think in the study, there were some like muscle biopsies done just to show, biologically, physiologically, there's an improvement in mitochondria in the muscle cells. So it is something, and it's something that we continue to research. We're trying to understand, well, how does this compound actually act? What seems to be slowing Alzheimer's? Really exciting, and like taurine, it's really a pretty safe supplement that people can.
It's quite safe.
Yeah, with Parkinson's disease, Another compound that seems to be efficacious is lithium, which actually you can buy a form of lithium over the counter, right? You don't have to have a prescription. People who have bipolar, lithium's been used to treat it. And the cool thing about that with the lithium is it seems to be efficacious. So again, I would always tell people who have been diagnosed with Parkinson or Alzheimer's disease to discuss even any supplements they're taking, to discuss it with their neurologist. But I think it's really exciting that there are safe and readily available, drugs that are possible therapeutics for these disorders.
Well, about the application or about the supplements, while listening to your share, many of our followers also have some questions to consult. May I convey their questions to you?
Sure.
Well, the first question is that what is the therapeutic potential of some popular anti-aging substances such as Rapamycin in the neurodegenerative diseases, or some common anti-aging methods such as caloric restriction? Is that beneficial for preventing Alzheimer's diseases? Or
yeah, it actually turns out that there have been studies in several Alzheimer's and Parkinson's models that including a publication from our own lab in a genetic Parkinson's model that suggests that rapamycin is something that is of benefit. Again, there are development of things called they're compounds that may be structurally related to rapamycin, but have less toxic side effect and or more effective. Caloric restriction as well. People like Mark Mattson at the NIA have done studies where they've shown that caloric restriction is something that helps in terms, or is possible, like therapeutic for PD and AD. I mean, one of the issues with its use in Parkinson's disease is people with PD, when it's sort of full blown, they actually experience a lot of weight loss. So I think what's important about caloric restriction is a lot of science has been used in the last decade or so to try to understand the mechanisms by which caloric restriction is driving these and its beneficial effects in aging and a lot of chronic age-related diseases, to see if we can come up with drug mimetics of that. One of those might be metformin, which is currently being assessed as well, but used in testing in older, otherwise sedentary individuals. So I think it's really cool, because I think it's kind of this advent of, up until now, we've kind of studied disease in silos, but over the last 15 years ago, scientists had this epiphany, realizing, wow, there's probably aging's not just a risk factor, but there are these common underlying processes like cellular senescence or mitochondrial dysfunction that are really contributing to these very different diseases. So if we can find a way to, for example, eliminate senescent cells, that's going to be useful in a lot of different age-related diseases. It's just making sure that we are not causing toxic side effects, and that's part of the game with
sports or other activities or sleep. How much an impact does sleep and other activities have on these neurodegenerative diseases?
So exercise, very important. As I had mentioned, with Parkinson's disease, doing high intensity, like short, high intensity exercise has been shown with Parkinson's disease. It's actually has a pretty amazing amazing effect. And so I think, yes, things like exercise, very important, and also staving off these other comorbidities that occur with aging that may contribute, like diabetes and et cetera. Sleep. This is something that's very, very important. And again, I think like the dietary restriction in the last decade or so, people realizing that maintaining your circadian rhythm and appropriate sleep is really, really important for your overall health when you're aging. I mean, ironically, people tend to have more trouble with sleep as they get older. But you know, things like exercise can help with that. Yeah, very important part of health.
And what are other diets or supplements are beneficial for preventing brain aging? Can coffee help when it comes to food or drink?
Yeah, it seems like the rules with this change all of the time, but certainly recent studies have suggested that coffee is beneficial, and other, you know, proteins, nuts, etc. It's interestingly, there have been epidemiological studies done, and drinking coffee actually has been found to reduce your risk of Parkinson's disease. It's one of the things that, you know, so coffee in terms of Parkinson's risk, consuming coffee reduces it. But it's probably sort of a bell-shaped curve with everything in moderation, because of course taking a lot of coffee can impact on your sleep. So you know, getting a dosage, getting a certain dosage. I think the recommendation now is like 2 cups a day, something like that.
Well, a rather professional audience has raised a question that recently the FDA have cleared lecanemab for entry into approval, which is the first beta-amyloid directed antibody to move from the accelerated to conventional approval. And are you optimistic about this drug? Do you think it is promising?
I actually do. I mean, I actually do think it's promising. We've had 20 years of clinical trials with these A beta immunotherapies, which have not shown improvement in cognition. With lecanemab, it's really the first one where it's been shown to not only clear the A beta plaques, but there's something like 20% slowing of cognitive loss. That the issue with it is, again, as I mentioned, this side effect when the antibody interacts with blood vessels or vasculature in the brain. This can cause swelling and brain bleeding. And it's not in all patients, it's a subset of patients, but that can lead to death. So it's a, you know, it's a pretty significant side effect. So that's something, if you are being treated now with lecanemab, it has to be done in a setting where they can monitor you for brain bleeding. So that's a little scary. But again, I think with targeted therapy where you're preventing this interactions with the blood vessels, and it's only eliminating the plaques in the brain. And I think we're getting there. Well, I am hopeful. And it, you know, it also suggests people, because of all these failed trials, a lot of people you know, said, oh, A beta, this plaque formation, it's nothing to do with disease. But the fact that giving this A-beta immunotherapy, where you're getting rid of the plaques and it's improving cognition, I mean, it suggests that A-beta does likely play a role in Alzheimer's. It may not be the only thing. There may be other things, tau, ApoE status, you know, how immunoaging
It's a really complicated question.
It's very complicated. But I actually have, I don't know, with the constant advent of all of these new scientific findings and technology, I think we are to a point where we can slow and maybe even reverse some of these age-related diseases. I don't think we're quite there yet, but I think we've gleaned a lot of clues. And I think a lot of this is going to be based on personalized medicine. It's going to be based on understanding what your genetic makeup is, your risk factors, looking towards your lifestyle, targeted therapeutics that can eliminate off-target effects. But you know, we've had, and I will say I am in charge of, or one of the people in charge of the PhD program at the Buck, and I see there's just a growing number of amazing young scientists that are entering the field and very interested in the challenge that aging and age-related diseases
it all set for us new hope for the promising future of the aging area.
Great hope for the future. I definitely, and I'm a bit biased, but I definitely think curing aging and age-related disease is really the next major scientific frontier. Really exciting, the breadth of, you know, so I feel like as part of this group of scientists, my interest may lie with particular diseases, but I think findings are likely to have implications across a lot of these different age-related disorders. So very excited.
I quite agree with you on that. This is also the goal and our detection of the Timepie, since that we hope whole body of human beings can aging slow, living well.
Yeah, absolutely. No, this is a wonderful thing you're doing. It looks like you have a enormous audience, and yeah, I love doing science. It's wonderful.
And maybe we come to the last one question. Before we prevent or before we began to treat diseases, what tests can be done to learn early about the neurologic aging? Through some games or any activities or detections in the hospital?
Yeah, I mean, there certainly are, for genetic forms of these disorders, there certainly are genetics tests, but also, you know, there are behavioral or motor tests that can be run at, you know, really in a way as part of like an annual physical. I think as I've gotten older, I think I am a lot more diligent about making sure that I keep track of markers of good, so I keep track of my blood pressure and my cholesterol and my blood glucose, all of those things. I think it's important for all of us as individuals to really take charge of our own health and insist that we have these tests run. So you know, I think that's one of the most important things you can do for yourself and your health.
And looking ahead for the future, do you believe that aging can be reversed, or can the Alzheimer diseases and other age-related neurodegenerative diseases
I mean, I think
be reversed?
we are getting to this place. So we start with kind of prevention, or we start at places where if you catch it early, but I think we are getting to a place where it's not outside of the realms of possibility with this the exponential progression of new scientific discoveries, I think it is entirely possible that we will get to a place where we will be able to review. I hope. That's the great hope of the field. So
we're also looking forward to the bright future of that.
Yes, that's right. Yeah.
Well, maybe, well, although not willing to, our interview comes to an end. It's really appreciate that you accept our interview and share with us so much detailed and fruitful interpretation on this topic about brain aging.
Yeah.
Well, I hope that everything goes well with you and we may get close contact in the future.
Yes. Thank you so much for inviting me. This is wonderful.
And wish you a good day and a good sleep.
Yes, thank you. You too. All right.
Bye.